The first drug manufactured using recombinant DNA technology with insulin, which is produced in bacterial cells, E. coli. Before 1982, people with type 1 diabetes mellitus obtained the insulin that they injected daily from pancreas removed from cattle in slaughterhouses. Cattle insulin is so similar to the human peptide, differing in only 2 of its 51 amino acids, that most people with diabetes could use it. However, about 1 in 20 patients is allergic to cow insulin because of the slight chemical difference. Before recombinant DNA technology, the allergic patient had to use expensive combination of insulin from other animal or from human cadavers. Insulin is peptide and is therefore straightforward to mass produce in bacteria. Some drugs, however, require that sugars be attached to peptide or protein, or that the protein folds in a specific intricate way to function. Transgenic non-human mammals can express human genes in their milk, including genes that encode clotting factors, clot cluster, and the connective tissue protein collagen. Human antibodies produced in rabbit and cow milk, for example, are used to treat cancers. Malaria remains one of the African deadliest diseases, killing nearly 500,000 children under the age of 5 and accounting for approximately 95% of global malaria cases and 96% of the deaths in 2021. Nearly every minute, a child under 5 years old dies of malaria. A vaccine that protects against malaria is based on the altering bacterium that normally inhabit the mosquito gut. Mosquito transmit Plasmodium falciparum, the parasite that causes malaria. The bacteria are given gene from E. coli bacteria that enable them to produce protein that tear apart the insect intestine. Using recombinant bacteria is easier than genetically modified mosquito to prevent malaria. Atlantic salmon that grow faster on less food than unaltered salmon due to gene from Chinook salmon and a gene from eel like ocean boat is an example of transgenic organism. Messenger RNA vaccine is made using messenger RNA that give your cells instructions for how to make the spike protein found on the surface of the COVID-19 virus. After vaccine, your immune cells begin making the spike protein and displaying them on cell surface. This causes your body to create antibodies that can fight the COVID-19 virus. A viral vector vaccine is made when genetic material from COVID-19 virus is inserted into unrelated harmless virus. When the viral vector gene get into your cells, it delivers genetic material from COVID-19 virus that gives your cells an instruction for how to make the spike protein found on the surface of the COVID-19 virus. Once your cell displays the spike protein on their surface, your immune system creates antibodies that can fight the COVID-19 virus. Subunit vaccine include only the parts of the virus that best stimulate your immune cell. This type of COVID-19 vaccine contains harmless S protein. Once your immune system recognizes that S protein, it creates antibodies and defensive white blood cells. If you later become infected with COVID-19, the antibodies will fight the virus.